Good morning, guys. It's Ms. Faye, and today we're going to be covering mitosis and cytokinesis. By the end of today's lesson, you should be able to identify what mitosis is, summarize the stages of mitosis, be able to recognize the stage of mitosis from a diagram, and explain what the role, or describe what the role of cytokinesis does, or what role it plays in both plant and animal cells, and describe how a cell maintains a diploid chromosome number, meaning how does the cell begin and end after mitosis, before and after mitosis, with the same number of chromosomes. And finally, what role does mitosis play in asexual reproduction, growth, and repair? By the end of mitosis and cytokinesis, and if you remember from the previous discussion, we said that mitosis is the division of the nucleus, while cytokinesis is the division of the cytoplasm. By the end of these two processes, what ends up being produced is two genetically identical cells. So basically, during interphase, the cell is preparing to divide. This happens before mitosis. If you remember, during S phase of interphase, or during the S stage, the DNA gets replicated, it gets duplicated, and then eventually, the reason why this happens, okay, the reason why this is important, that the DNA gets duplicated, is so that at the end of mitosis and cytokinesis, when the cell splits into two identical daughter cells, the DNA that was present in the original cell basically gets duplicated, so that half of that duplication goes in the first daughter cell, and the second half of the duplication goes in the other daughter cell, okay? Because if the DNA was not duplicated, then you would not have enough identical DNA to go to two daughter cells, okay? So that is the significance of duplicating the DNA during interphase. So mitosis divides the cell's nucleus in 4 phases, or 4 steps. And the first step that we want to focus on is prophase, okay? And just like it says here, during prophase, what ends up happening is that the chromosomes condense, and these structures known as spindle fibers form, okay? And spindle fibers are basically, if we go back to the beginning when we were talking about organelles that are found in the cells, spindle fibers are made from microtubules, which means that they are made from that those organelles, the protein fibers that we discussed at the beginning of the year when we were talking about organelles that are found inside cells. And the spindle fibers, so these things that you see right here, these are the spindle fibers, they're going to be very important because they're essentially going to attach themselves to the chromosomes during this process, during the process of mitosis, in order to help the chromosomes separate from one another and then eventually make their way to the two new cells, to the two daughter cells, okay? Another thing that happens during prophase, which is the first stage of mitosis, or the first step, is that the chromosomes become condensed. They become thickened and visible. And if you remember, when I was, during our last lesson, I said that in interphase, at the beginning, before this whole division happens, the chromosomes are so thin, they're barely visible, and they almost look like spaghetti or noodles, very, very thin, okay? They're not condensed. We can't see them at that stage if we were to look underneath, at a cell underneath a microscope. During prophase, when mitosis begins, the chromosomes, they become visible, okay? So they become kind of like condensed and we can see them. Okay, next is metaphase. And during metaphase, I want you to remember M stands for middle, okay? Metaphase, middle. What happens is those chromosomes that became condensed line up, just like you can see in this image, they line up in the middle of the cell. They line up in the cell's equator. And as you can see, the chromosomes lined up in the middle of the cell with the help of who? The spindle fibers that we just talked about in the previous slide. So these spindle fibers that you see here, these structures, basically these lines, they attach themselves to the chromosomes and they help to kind of move them around, and they help to move them into the middle of the cell during metaphase. So metaphase, chromosomes line up in the middle, in the equator. Next, we have anaphase. Okay, so the next step is anaphase, and during anaphase, what ends up happening is that those chromosomes that were in the middle of the cell start to move away from one another, okay? Away from one another. And you can remember it because anaphase begins with the letter A, and so does the word away. Chromosomes move away from another. Who is helping the chromosomes move? Again, we can see the action of the spindle fibers, right? So, oops, sorry. We can see the action of the spindle fibers right here, okay? So the spindle fibers are helping the chromosomes move away from each other. And they do that by attaching themselves to the location in the center. If you remember what this is called, the center of the chromosome. So if we look at a picture of a chromosome, the center of the chromosome is known as the centromere, correct? And this is basically where the spindle fibers, centromere, and this is where the spindle fibers are basically going to attach themselves to, and then they can actually move the chromosomes. All right, next we have telophase, okay? And during telophase, what ends up happening is after the chromosomes have moved away, okay, from each other, two new nuclei form, okay? Two new nuclei form. One around the first set of chromosomes, another one around the second set of chromosomes. I forgot to mention this, but during prophase, the first step of mitosis, the nucleus breaks down, it disappears, and it does that so that we can, so that the chromosomes become exposed. We can actually see the chromosomes, all right? So obviously, if you break away the nucleus during prophase, during telophase, what ends up happening is you end up forming two new nuclei, one around each set of chromosomes. Another thing that happens is that those thickened chromosomes that we saw and that formed in the beginning in prophase now begin to uncoil, meaning they become less thick. They go back to that noodle-like formation that they looked like at the beginning, meaning that they are not visible. They're not as visible. They're not thickened. They're not condensed as before. You'll also notice that those spindle fibers are starting to kind of retract, and they go back to the organelles that help form them, or that are associated with them, which are the centrioles. And we know we talked about centrioles before, and we said that they play a role in cell division, and this is the role that they play. They kind of work with the spindle fibers to help the chromosomes move. Finally, we have cytokinesis, and cytokinesis is defined as the division of the cytoplasm. So once you have the two nuclei forming around the two sets of chromosomes and you have the two daughter cells must basically separate. Okay, so the cytoplasm essentially splits and you end up with two brand new cells, two daughter cells. The division of the cytoplasm, that is what cytokinesis is. And it differs a bit depending on plants and animals. In animal cells, something known as a cleavage furrow forms, and that is what basically causes the cytoplasm to split. That is where the cytoplasm splits. It splits at this location known as the cleavage furrow that we see right here, okay? I don't know why my pen likes to be weird. Okay, I'm just gonna give up. It is going insane, forget it. So essentially the cleavage furrow, I'm just gonna use my MacBook instead. This area right here, I hope you can see it. So this is where the cytoplasm is gonna split. In plant cells, something known as a cell plate forms. Remember, plant cells, they are usually found one right next to the other. They don't really separate, okay? So in order to separate them, because they can't physically split like animal cells can, like here, they physically split into two distinct cells. In plant cells, they remain next to each other because plant cells need that kind of like extra support, and they also have the cell wall, okay, that is present around them. So therefore, in order to accommodate for that, something known as a cell plate forms, which basically creates two new cell walls around each cell, and therefore separating them. They're not physically separated, like they're still right next to each other, but at least they have that division, that wall, that gate, or whatever you want to call it, between them, which is the cell wall, which formed due to the action of the cell plate. Okay, now so we know that mitosis plays a very important role in our bodies because it creates identical cells. It creates these daughter cells, which are going to serve so many purposes, including helping us grow and develop, but also repair when we do have a damaged tissue or damaged cells and we need to replace it with new cells. Mitosis is the way, and cytokinesis are the way to do it. They produce new cells to replace old cells, to replace damaged cells. Another reason or another significance of mitosis in prokaryotic organisms, it is actually used as a form of reproduction. So asexual reproduction can occur by mitosis, specifically a process known as binary fission, which produces two identical, genetically identical daughter cells from one parental cell. And a perfect example of an organism that uses this form of reproduction, of this form of asexual reproduction, are bacterial cells. And bacteria use binary fission to reproduce, and that is why they are able to reproduce so fast, okay? That is why you can end up in 10 hours, or in 10 cycles of, or 10 division cycles, just like this graph shows right here. In 10 division cycles, you can go from 0 or very little bacteria all the way up to around 1,000. Okay, so that's a very short period of time. And they can divide super rapidly because they use mitosis, and because they're just so simple, and they don't have that much complexity in their structures in order for them to take a long time to reproduce. So all they do is basically take an original parental bacterial cell, and by using binary fission, which is pretty much exactly like mitosis, it divides into two daughter cells, identical daughter cells. There's no genetic variation there. Another example of organisms that reproduce through mitosis is this organism right here, which is a hydra, but also yeast. So both these organisms are eukaryotic. They're not prokaryotic. However, they still have the ability to divide through mitosis, meaning that they produce identical cells. They start with an original cell and they end up with identical or two identical daughter cells through mitosis. Finally, we have two other eukaryotic organisms that reproduce by mitosis. Here we have an example of a process known as fragmentation, which occurs in several organisms, but like the classic example is the starfish. And what ends up happening is if the starfish is split, like the arms of the starfish are split, each arm will develop into a brand new organism, into a brand new starfish, using the process of mitosis. This is known as fragmentation because you're essentially taking that starfish and fragmenting it, cutting it into bits, and that bit or that piece grows into a brand new organism. Vegetative reproduction occurs in plants, and I'm sure you've seen this in action. There are certain types of plants where if you take a cut from one plant and just place it in water, you end up with a brand new plant. Okay, so this plant that you took from the original mother plant is going to be an identical copy, and therefore this type of reproduction occurs with mitosis. Now, what is the advantage of reproducing asexually using mitosis? First of all, like we saw in the bacteria, it's very fast, okay, very, very efficient. It's not very complex. And if the conditions are favorable in the environment, you can end up with thousands to millions of offspring of daughter cells in a very short period of time. However, the disadvantage is all of the daughter cells that are produced during asexual reproduction, during mitosis, are going to be identical. So if that original parent cell has something wrong with its DNA, okay, some damage happened to its DNA, all of the daughter cells are gonna also have damaged DNA. There's literally no room for genetic variation, okay? Sexual reproduction, although takes longer and it's more energy consuming, it does produce a lot more genetic variation. And that's why we see so much genetic variation in our populations, because we reproduce sexually, and so do many other organisms on Earth. So although they do have their advantages and disadvantages, it really depends on the organism, and it really depends on the environment and the environmental conditions. And that will determine whether or not asexual or sexual reproduction will occur in some organisms. And also, that will determine what are the advantages and what are the disadvantages. Okay, that's about it for me today. As usual, if you have any questions, please let me know. Have a great one.